Hello, audience. Can you hear me? Just put it in the chat if you can hear me. Yes, perfect. Okay, thank you. So, welcome everybody. Welcome to the first tutorial in this online form. Very exciting to do this. It's the first time we do this and a new experience, I think, for many of you. So we start with a tutorial, but before I started that, I would like to say that if you are not happy with the screen you see, in the right bottom there are 4 small views visible, and you can click on one of those to change the size of the presentation that you see. If you have questions, put them under the question mark. I will, during the tutorial, look at the question marks regularly. I will note the questions, and after the presentation by Christian, I will give the floor to the people that ask questions. So the presentation, I understand, takes a little bit more than 1 hour, and that means that we have ample time to discuss what Christian has discussed. So don't worry, questions is easiest if you ask them during the presentation in the question mark. Okay, it's not about me, it's about Christian. So I didn't introduce myself. My name is Mark van Zandvoort, but now you know. And then it's the floor to Christian, who will give a presentation on localization microscopy. Maybe, Christian, you can introduce yourself and start your presentation.
Okay, thank you very much, Mark. And thanks for the tech support. So until now, everything works perfectly, no problems. And yeah, as Mark said, the presentation is not going to fill up the 2 hours. And at the end, we can decide if we want to have a discussion, which I think can be useful, or I have a couple of demos that I can show you on how to analyze a few things that I will show you during the presentation. And we can decide on how to proceed. And okay, yeah. So my name is Christian. I am a junior group leader at HZI, which is the Helmholtz Research Institute for Infection Biology in Braunschweig in Germany. And we actually study virus infection. I'm gonna show you a few of my own things on these slides, and I have also another presentation later during the FOM week. But this tutorial is really going to be a tutorial, so sort of a basic introduction into localization microscopy, with some theory and some history. Actually, now it's already been a while in science. These techniques, and then it's going to be a lot about practical considerations, what you might have to think about before or during working with these techniques. Case studies, I think, are super important to get an idea of what you can do, especially since I'm sort of promising you insights into nanophysiology. So let's have a look at that. And at the end, as I said, I can show you a few analysis steps to get you started into some quantification from SMLM data. So just since you are sitting in front of your computer, I am sort of maintaining this GitHub wiki, which was actually thought to come along with this tutorial. So it's supposed to be an introduction into SMLM, a few introduction points, and then a lot about methodologies that I have mostly used myself and that I want to maintain for me and for like students and people working with me. So right now, I think it can be useful for you if you want to browse this website. And I'm also going to come back to that every now and then. And at the end, we can discuss or go through 2 of these points that are shown on the right, 2 of these specific, let's say, processing points. Okay. All right, so let's get started. And just to mention, I cannot see you right now. And as Mark said, if you have any comments or you cannot hear me or something, just put it into the chat or raise your voice if you can. Okay, so let's start with fluorescence microscopy. or you may not know, but in fluorescence microscopy or in simple bright field microscopy, we really don't have a lot of information about specific structures that we look at. We can maybe identify a few things from just putting light on the sample. Like here, maybe we can see the nucleus or anything that makes contrast, or we can see a few granules, but that's really about it. So the power of fluorescence microscopy, very, very simply speaking, is to put contrast on specific molecules. We can put that in. We can, of course, combine labels that are spectrally different and all these type of tricks. But in general, we can make things visible that you can't with just light. Now, the problem with standard microscopy, so to go towards resolution and magnification now, is that okay. So this is just summarizing the pros that I just mentioned. Sorry. So we can separate different structures. In this case, we can separate the cytoskeleton from the nuclear DNA. We have high contrast and high specificity. So the contrast, the problem with this. With resolution at this point is that due to the diffraction limit of light, we have the so-called Abbe limit, which is about half of the wavelengths. So it's typically around 1 to 300 nanometer. So, which means that if we look at microtubules in this case, they will not appear as small as they are or as they could be, but they will be around 200 nanometer, let's say, in diameter. And that has been a well-defined and well-known, limit. It's not a problem, really. It's just a limit of conventional microscopy techniques known for a long time. So now towards localization microscopy. So the point of localization microscopy is to isolate molecules, and we can do that since we have a very good understanding of how the microscope, which is shown on the left, really interprets a single molecule. And this is really based on the optical configuration of the microscope. So if you image an individual molecule, what you see at the end with your eye or with your detector is a so-called point spread function. So the information, the emitted light of the point from the individual molecule will be sort of accumulate into this egg-shaped point spread function. You can also look at that, using this PSF generator in XY, and you see that it's sort of this wavy pattern, which is called an airy disk. You can look at it from the side, and you see this sort of egg shape, maybe in the center, that then spreads because of the focusing of the objective. And that's basically one of the most important concepts in localization microscopy, that we have a good idea of this point spread function. So we can identify, or we can just put a line across this point spread function, and we see that it has a width of about what I said before, about 300 nanometer in this case, and that depends again on the wavelength, but that's the fundamental limit of what we can see. Then this is just a single emitter. And of course, the problem gets, or this limit gets even more visible if we have multiple emitters. Let's say they come close together. So if there are 2 points, of course, we can say, yeah, we can really nicely localize those. But if they come closer together, we lose that limit, and they somehow, at some point will merge into one. We don't have any idea of what is, if it's 2 or 1 and how far they are apart. So yeah, the trick in localization microscopy is to go back and to avoid this overlap and to have individual molecules in your camera frame, let's say. So the general workflow is always that we try to photo switch, or we try to make molecules, we try to separate them so that you have this two-dimensional point spread function. Here, this is an actual example here on the top. Then we perform a two-dimensional Gaussian fitting in the easiest case. I will mention a few words about that later, which, since we have an idea of how this point spread function looks like, we can pinpoint the localization of this underlying molecule, or we can pinpoint the center of this point spread function at a much higher precision, typically, of course, below the size of a pixel. So we have sub-pixel resolution. Now, the precision of how well we can define that position depends on the number of photons. So it's correlated to the number of photons, which is this n number here, and it's simply kind of would raise the height of this peak, and of course it makes it then easier for the fitting to find the center. Okay, and at the end, typically of one of those cycles, these molecules will disappear again because we don't want to have them visible for too long. Otherwise, they would overlap at some point, or we would just not image many of them. So at the end, they would bleach or photo switch back, and then we can start another cycle. Okay, so then I would say the three typically used methods in SMLM. So there are many acronyms and there are many different variants of SMLM. That's why I'm a bit hesitant now. But I would say these three that sort of are most common, maybe, or most widely used. So they are summarized as STORM, stochastic optical reconstruction. Then we have photoactivated localization microscopy, and these were the first two that somehow became popular. And actually, together with these, point accumulation and nanoscale topography, paint was also developed. And I will show you a bit of the history of that. But these techniques really have come a long way and are now really capable of doing many great things in nanoscale biology. So to visualize this process again a little bit more, so we have a target structure that's labeled with a fluorescent dye or fluorescent protein with fluorophore, and since they, due to the diffraction limit, we have a certain width of that structure, and all of these molecules overlap. But we can apply this trick, so of making them separate from one another, which sort of looks like this. This is obviously not the same structure, but you see that we have these individual blinking events. And those events can then be localized, and we can pinpoint the localization or the center of these at a much higher precision and put all these localizations, which are simply XY coordinates, together to reconstruct a super resolved image. So importantly, for SMLM, the images that you see, they are always reconstructed. So they're not real images, which is different like compared to other super resolution methods, which are directly sort of producing a pixelated image. In SMLM, there are always reconstructions, which is important to know when looking at them and also when making them. But so the principal type of data is a list of localizations, like a table of XY coordinates. All right. So for Palm, so for when using photoactivatable or photoconvertible proteins, so we have really a range of different proteins available right now. Let's say I usually like these ones that turn from green to red, so they're photoconvertible using UV light. For example, amnios is quite popular, dendra. But then we have another class of proteins that are dark, so they don't emit. And upon photoactivation, they turn into a specific wavelength and can be visualized using your microscope. And that's dependent on the conversion within the chromophore. And it can, of course, be used not only for super resolution imaging, but for all sorts of other things, for tracking. I mean, they're quite popular for measuring diffusion within cells, or measuring long-range transport within neurons, let's say. So they're quite nice actually for many different things. And at some point, I suppose that happened. I don't know, that someone realized, look, we can perform sort of nanoscale imaging with these fluorophores by just looking at individual copies. And that's when Palm was introduced. And this is really this is a figure from the original paper, and that was performed on fixed cells. And you can see here, this is a sort of a representation of this activation, imaging, and bleaching cycle that is then accumulated for a number of fluorophores or a number of cycles until you have your final image. You can see, of course, that the resolution increases compared to the wide field reconstruction, or the wide field image. They were able to reach a localization precision. Maybe you saw this in one of the earlier slides, that this precision with which we can determine the center of a PSF is called the localization precision. and it's abbreviated sigma often, so you might see this again. And they reach something about 24 to 60 nanometers here. And yes, this was performed in fixed cells, and they also had to do, I think, a correlative EM study to make sure that these molecules are really labeling this type of structure. Okay, so this was the beginning of PALM. Here I'm showing you, this is a video that I made of a cell culture. The cells were transfected with an EGFR connected to MEOS, and I'm just moving around with having the 405 laser on and imaging in the 561 sort of channel using 561 nanometer excitation. And now the video is almost over. Let me go back. So, you know, when I move that activation laser spot on a cell, you can see that we see an increase of these localizations. You know, they start blinking. Just to give you an idea of how this type of data looks like. So it's for me, it was also a very good start to get into localization microscopy, because, you know, if you do transfections or something like that regularly, you can just transfect your favorite cell line with a photo convertible activatable protein and then just start doing SMLM. So that's quite a nice success story. Okay, so let's mention a few pros and cons about PALM, or of using photo activator photo convertible proteins. Of course, they are very specific because you have a genetic conjugate and you have ideally full labeling, so all of your proteins are labeled. That's of course not entirely true. Because there are maturation effects that might lower your that might lower this labeling efficiency. You of course also depend on expression level. So typically, or what is like the most straightforward thing to do is to have an overexpressing plasmid, as in this case, and that is not going to represent your endogenous protein population. Another advantage is that you can do live cell imaging. So this was a live cell sample. You can perform tracking. I will come back to that later. But usually then mentioned in the same sort of connection is that you might, of course, change the dynamic behavior of your protein by putting a fluorescent protein on it. So that's something also to consider. I would say it's easy to implement, but okay, that's something for me to say. But as I said, you can just transfect your plasmid and perform a PALM experiment. The signal to noise, I will show you another example of another technique then, is typically not as high as with other localization microscopy techniques, which affects the localization precision, as we said before. Okay, so then in Storm started like this actually. So they used a dye pair on a DNA origami structure, like a very simple origami structure, and then here you see this you know, a different representation of this activation localization bleaching cycle. So in Storm it's a little bit different, is that we have all the molecules on in the beginning, and they need to be put into a dark state. And then from there, they stochastically return to the on state, and they are imaged or localized, they bleach, so now they are dead. Then comes another one, and so on and so on. And in this case, they use this readout dye together with an activator, so an activator reporter dye. So they have this sort of a bit more complicated scheme of readout, activation, and so on. So this was the initial version of Storm. Now, why does it blink? So it actually depends on at least two components within the buffer. One is the presence of thiols. So here's it's a simple Jablonski diagram of these different states. So you have your transition to the activated state from the ground state of the fluorophore. From there, the fluorophores can go to a so-called triplet state and either go back directly to the ground state, or they can also bleach. So there's one version that's not shown here. But what you want is that you transition from the triplet state to the off state, it's called here. So it's sort of a reduced state of the molecule, and that is this long-term dark state. So these molecules stay off for seconds. So all of these different states have different lifetimes, which is also very critical. So the excited state So these molecules, this is a very short-lived state, and the molecules return very quickly to the ground state. So I think it's a matter of nanoseconds even, and that's something that is measured in, for example, lifetime measurements. So these molecules cycle in this very first part for I don't know how many, a few hundreds or a few thousand cycles, and that's when you harvest your photons. From there, every now and then, all of these transitions have probabilities. The molecules might transition into a triplet state, and from there, if you have the right buffer, they can go into the off state and just stay there. So then you ideally have this situation here that all of the molecules, or let's say most of them, are dark. And then from there, they either turn back stochastically, or you can use an activation laser to bring them back to sort of have a better control of the density of blinking events. And sort of figuring out this entire these properties for small organic dyes, I think, was the big point of the early days of STORM. So there was a further development of this type of STORM, which was later called direct STORM, because it doesn't actually require this activator dye. You can do it with just Cy5. Again, using the same principle as shown here. So with same buffer component. I forgot to say that usually there's an oxygen scavenger system in this buffer that prevents bleaching of these dyes and prevents the reaction of the triplet state with molecular oxygen. So these are typically the two components of the switching buffer. It's some sort of thiol and a molecular oxygen scavenger in a pH buffer. Okay, so then a bit later, some people figured out, look, it also works with just Cy5. We can get molecule blinking. We can isolate individual molecules at quite a high localization precision. And I'm saying this because I think nowadays probably most people use the direct STORM version. Okay, so you can already tell from these early papers that STORM reaches a higher localization precision because there will be multiple factors. It's just that you have a different environment. So typically the cells are fixed, so you can use higher laser irradiance. You use small organic dyes, which produce more photons. So all of these factors together lead to a lower localization precision. I can also show you an example, which I had shown before. So maybe you can already, by looking at this example, appreciate that in STORM, using Alexa 647 now, which is a very popular STORM dye, you can reach a higher signal to noise, and you can have, which translates into a higher localization precision. So that directly goes here into my little table. So they are very bright, and you have a high signal to noise, which leads to a higher localization precision, not a higher resolution necessarily. Another advantage is that we have, we use, or typically we use antibodies to label structures with these small organic dyes. And that means that we can label the endogenous population of proteins. So it doesn't require any cloning, and we can look at the level of the natural, let's say, sort of expression level. And disadvantage of that, of course, is that we have to chemically fix our cells, which might produce other forms of artifacts, or might not be desirable for all experiments that you might want to do. Okay. Yes. Okay, so then paint was the third method that I wanted to mention, as it I think is quite popular now also in life sciences. Paint started with imaging vesicles actually. So in paint, the principal idea is that you have a transient interaction of a labeled structure. So it doesn't depend on photo switching of a dye that is in place, but it depends on a transient interaction with a fluorescent dye. In this case, they used Nile red, which is a fluorogenic dye that interacts with membranes and then lights up and then leaves again or bleaches. And so already very early, even before Palm and Storm came up, it was possible to perform localization microscopy using Nile red sort of photo switching or transient photo blinking. So that's quite nice actually. And these are the images from that original paper. So here they looked at vesicles. I don't know, probably something like a small unilamellar vesicles. So like 100 nanometer, and this is a patch of a membrane. I think so. It's no cells or anything. So this is, I think, a planar bilayer. And they were able to perform localization microscopy on these using NIRED. NIRED is still also used to image, for example, bacteria. Here's also, I think, this is one of the first examples where bacteria were imaged with this with this technique. And the reason why you see two dots here everywhere is that they used a different shaped point spread function, which is called the double helix. And these are simply the two lobes. So for each localization, as we would have seen it before, as we see here, maybe in this case, you see two lobes, and the angle between the two gives you an idea about the axial position. But I'm going to mention this later when I come to multidimensional imaging. So this was the beginning of paint. I'll come back to like more other implementations of paint later. Okay, so this was sort of the theory and the history of these techniques, and let's go back to the future now. So I want to show you a few things that we can do or that we do nowadays with these techniques. And while I do that, I want to mention a few practical aspects that are, I think, important to consider when working with localization microscopy. And we'll go through all of these. So localization blinking, you might have, you can expect, or you might guess that that the blinking can be an advantage, but it can also be a disadvantage and needs to be taken care of. I want to talk about resolution and then multidimensional imaging. Okay, so localization is sort of the first part or the first analysis step when generating localization microscopy data sets. And there have been many different softwares now developed to perform this step. And at this point, you can, for example, go to this to the SMLM tutorial wiki and click on the software tab, where I try to summarize a few options that I used myself. Of course, it's very biased, but I think they are useful and they are easy to use, and they might, they just get you started. And some of these are shown here. So Thunderstorm, I think, is quite popular. It is already 11 years old. So it's an ImageJ plugin, and it really became very popular because it's quite easy to use, quite intuitive, and you have a visual readout of your localization procedure. Picasso and SMAP are more recent ones. Picasso is a Python-based localization software, and SMAP is a MATLAB-based software. And they are a bit more sophisticated, let's say, and have other options and allow you, like, have GPU support, which is necessary nowadays for many things. And these are sort of the softwares that I use a lot myself. But if you want to know more, so there's an entire sort of development community, I think, and they all came together during, I think, 2 or 3 localization microscopy challenges, which can be found if you just Google localization microscopy challenge. I didn't put the link here, but maybe it's on the wiki. It's hosted on a site from EPFL, because it was initiated from Daniel Sarge and a few others. And there you can find like data sets. If you want to work on your own software, you can find simulated or experimental data sets to challenge your own software, and of course a list of available softwares. And there's really a lot. So and they require different expert levels, different programming skills, and all of that. So the list that I showed you on the previous slide and also on the wiki is again very biased to what I have used myself. And you're free to explore these sites. Just one more recent localization software that I want to mention, because it seems to be superior to actually all of the different variants that have been published before. It's called Decode, and it uses that trains a neural network to identify localizations. For me, the intuitive explanation of why it is so good is that, of course, it can be trained on many different density regimes, and the density is always something that is critical, because if you have an individual molecule, all of these softwares, they already perform very well on a, let's say, on a two-dimensional point spread function. But if they start to overlap, or if they have different signal to noise ratios, then you see them split apart. So then some have advantages while others have not. And here you can see that in this paper, they produced different data sets, and they compared this decode algorithm with some others using two-dimensional data, astigmatic data, and double helix data, which is 3D. And it seems that in all of them, the localization error as well is low. And the detection accuracy is high. So it seems to be quite superior. I haven't used it myself, but I still think it's like, if you start SMLM now and you don't have much experience, it's probably a good thing to keep in mind, because it seems to be very good. It seems to be very precise. Okay, so let's start about blinking and counting, because they are somewhat connected. So one sort of observation that was made for SMLM is that molecules typically appear more than once in Palm and in Storm. In Paint, it's not so much of a problem, as we have a different mechanism of photo switching, as I said. So we have this transient interaction of individual molecules. So we don't really look at the same molecules for a long time, but they sort of exchange. So, but if we look at a Storm dye or a Palm fluorophore, we would see something like that they appear multiple times, which is simply cycling between the on and off states, which produces multiple localizations that, because it's always, for each localization, we perform a fitting step, it might not produce the exact same result. So there's some sort of uncertainty. Here's a chymograph of how this might look like. So for each localization, we have these so-called, blinking trains, or just localizations that come frame after frame. And here's one, here's another one, and so on. And that needs to be taken care of, for example, using a grouping or so-called merging analysis step, where these blinking events are merged into a single molecule position. So that is like going from localizations to protein or molecule positions. And of course, once we can characterize this sort of blinking, we have a much better idea of how many molecules we have in our data set. So it's sort of a counting operation. And the way to do this, to calibrate this, is by using a test sample. And that's something that I also describe in the wiki. It's basically a sample that does not have, or that has a known organization of your fluorophore. In the simplest case, it's just individual dyes, individual antibodies, let's say, or individual dyes. That is sort of the minimum organizational unit that you would expect in your sample. You can also use like more complex structures, like origamis that have 2, 3, 4. Like any known organization would suffice here. So I want to discuss this with a simple version where I look at one molecule. So if we look at the localizations from one molecule, as I said, they are recorded over different frames, and they can sort of scatter around the true position of that molecule. And by measuring the spread of this localization cluster, you can get the localization precision, which is the experimental localization precision sigma. And you also get the gap between the different localizations, or between the different blinking events, and you can use all that to correct this cluster into a single molecule position later on. So it's then corrected, and it can be used for counting operations. What comes out of this calibration is, again, the sigma, the localization precision, as well as the dark time histogram. And these are the two parameters that you would put into a grouping operation. And you get, so this is a dark time distribution. So these molecules can be dark for quite a long time. So then you have to sort of choose the dark time you want to set. In this case, or I used to put it on a very high value to have a 99% sort of probability to really capture all of the blinking events from a single molecule. So here in this case, it's really a few, like 22 seconds. But with this long dark time and localization precision, you really can correct your data and go from localizations into molecule positions. And that's something that I could show you later in a practical demonstration. Yes, it's there. Okay, so now let's talk about resolution and localization precision a little bit, because they're sort of mixed sometimes. And as I said, the localization precision is the precision with which we can localize an individual molecule, and localizations will scatter around the true position. And by measuring the spread, we can identify the experimental localization precision. Now, resolution is different. So resolution, I'm just showing this image to make this clear. Resolution really depends on the spatial arrangements of label that we have. So there's this Nyquist criterion that says, in a simple one-dimensional case, we need 2 labels per 1 unit of resolution. And that becomes clear when just looking at this image. So here they have this synthetic data of these squares, and if they are fully labeled, we can see, look, this is a square, but if we go down with the localization density, you see that you progressively lose that information. So that means that to have a certain resolution, we really need to reach a very high number of labels on our sample. And roughly speaking, of 1 point spread function, which is this x. So if we want to resolve 1 point spread function at 10 nanometer resolution, we need about 2,000 molecules, which is quite high. Now, if you think of one localization, so within that one localization, we would need we need to localize something like 2,000 molecules. So labeling is really critical. Otherwise, you lose a lot of information. So how can we measure resolution experimentally? If we don't know, we have a sample in front of it and it looks nice, but how can we know what the actual resolution is? So what many people do or have done in the past is to look at microtubules, as we know how big they are, how wide they are. So we can label microtubules and just try to see the double. So since it's a hollow tube, if we look at it in the projection, we would see 2 lines. And these 2 lines would have a diameter or a distance of 25 nanometer, which is just extended by the size of the label of your antibody complex. And that's already a good sign. If you can see the 2 lines and they are separated by, let's say, something that would make sense in terms of a microtubule together with 2 antibody complexes, let's say on both sides, which can go all the way up to 60 nanometers. So that's already a good measure that you have, at least on that scale, enough molecules to have a resolution sufficient to see the double peak on microtubules. So that's, of course, very much dependent on how you put this line plot. It's done with a line plot, or it depends on a couple of factors. So there are more general ways of measuring the resolution. One is called Fourier ring correlation, which is used in electron microscopy as well. There's a 3D version of this that's called Fourier shell correlation. And the point of FRC is to put or to separate your data set in 2 halves and make this Fourier transform, and then compare these 2 halves and see at which point they lose correlation, and that goes into the noise of your background. And that's sort of the resolution. So that's sort of, for me also, it used to be, it's not as intuitive maybe as the microtubule, but gives you a way to estimate your data set and compare it with other data sets as well. So that's also, it doesn't, I would say, it doesn't necessarily mean that you can expect everywhere on your data set a resolution corresponding to this number that comes out of the FRC FRC routine. It's just for me something, practically speaking, to compare your data set and also to compare different areas of a single data set, and this type, and maybe also as a filtering parameter. Okay, there's another development here that was published just 2 years ago, which is parameter free. I don't want to go into the details, but just go into the GitHub site. It's a ImageJ plugin, I think, and you just have your image open, maybe from some other analysis routine like Thunderstorm, and you can perform this resolution estimate by using this plugin. Okay, so importantly, we have seen this example for the microtubule. So the label is, of course, critical, and that's something that is now very well known, and most people have it in mind. So the antibody complex is still sort of the standard, but it's also one of the biggest affinity probes that is available there. So nowadays, many people try to go to smaller variants, either fragments or other antibodies like nanobodies or monobodies like synthetic affinity binders, or lots of other things. It's just important to keep in mind that your label will convolute the size of your structure. Here's an example from a nanobody against tubulin, and you can see that if you label these microtubules with a primary-secondary complex compared to a primary and then compared to the nanobody, you go all the way from, let's say, 60 over 50 to 40. And as I said earlier, we have about 25 for a microtubule. So that's really something to keep in mind when you make these estimates. Here's another more recent example of these small affinity probes, just by looking at individual points, a large primary-secondary complex compared to a small, let's say, alpha tech and a nanobody, and even a smaller extension of just a DNA fragment. You can go all the way down from 8 to 4 nanometer from a single point. So you might say that this doesn't really matter much as long as they are independent, but it makes a big difference once we look at a combination of labels or of labeling sites. And this was done here using, or nicely visualized, I would say here, using an origami array, an origami structure. So you have this 2x2, and you can maybe identify these 4 sites using an antibody, but you can not do it with 6 sites or even more sites. So you need smaller labels for that. Okay. So let's talk about multi-dimensional imaging before we go to the examples. So one dimension is, obviously, since we use fluorescence, is to perform multicolor experiments, because in fluorescence microscopy, that's usually one of the first things to do. But it's sort of limited because we have a limited spectral range for fluorescent proteins. On the other hand, we have a wide range of synthetic dyes, but they might not switch as well as you might want them to. So that's sort of a few things to consider. In live cells, we have some options, for example, combining different photo-switchable or photoconvertible proteins, as in this case here, PA-GFP was there combined together with PA-TAC RFP. So we have a number of these photo-switchable probes that can also be combined. We have also photo-activatable dyes, which is quite nice. So those were developed at Genelia Farm, and they can be combined with something like a snap or halo tagging of your protein. So you can put a photo switchable dye into a different spectral channel. There's another way. There's also another way of converting fluorophores, which is called a primed conversion, which gives you a bit more flexibility in terms of the spectral channel of your photoconvertible protein. So that works for a number of photoconvertible proteins. An easy way, this was the traditional way of doing multicolor imaging in STORM, was to combine different reporter activator pairs, which also was shown to result in very nice multicolor imaging. And the good thing is that if you use the same reporter and different activators, is that you have a good control over the photo switching, and you can use the same buffer. A disadvantage, in my opinion, is that it's quite extensive, or it requires a lot of work to label your antibodies with 2 dyes in the right ratio and so on. And there's also they're also prone to crosstalk, of course, because you use the same dye, the same readout dye, and they might have overlapping or might have cross-talking activators. What is, I think, becoming more and more popular is to perform ratiometric imaging for multicolor. So now this is in fixed cells, where we use the same spectral channel or a spectral channel that are very close. Let's say not the same, obviously, but they are very close, as you can see here. So this is Alexa 647 together with CF 680. So they are quite close and they overlap. So you can image them side by side or on two cameras, but it's often performed side by side on the same chip. And so you localize all of them, and by comparing the intensity on one side of the chip with the other side, you can identify the molecule, the identity of the molecule dye. And there's very many nice examples. So it's used by a few labs now, and I think performs very well. And it can even be extended to more than 2 dyes. Also, another way to perform multicolor imaging is by using DNA paint. So I haven't mentioned this earlier. So this is a different way of paint imaging, not by using like a fluorogenic dye, like in this Nile red case, but this is a more general procedure by using DNA oligos. So it's shown here. So you have a labeled DNA oligo that binds to its complementary oligo on your target structure, the P1 pair, and then you have a P2 pair, which is a different antibody or different structure that has a different oligo. So this is this is really possible to highly multiplex. So you can basically have an infinite, or theoretically have an infinite number of structures that you can image by washing out the by P1, putting in the P2, and so on. So this is a very good approach to perform multicolor imaging. You can also use the same detection channel, which reduces aberrations and these type of problems that might come up. So that's something that is that is another advantage of for multicolor imaging. An easy way also to perform multicolor imaging is by combining techniques. So for example, by combining Palm and Storm, as many of the Palm fluorophores work in Storm buffer. You can also combine Storm and Paint, as some of the Storm switching works in Paint compatible buffers and so on. So that's also something to consider. Here's another example of combining techniques using a primed conversion Palm together with a normal conversion Palm. So you can see two fluorescent proteins or photo convertible proteins together with, let's say, a paint and even a fourth modality. So that's something that also I would always recommend to keep in mind. So just a few words about co-localization. Co-localization is measured differently in SMLM. So you don't compare pixels, as we don't have a pixel image as the primary data. You can still do that, of course. You can just render an image and perform a Pearson correlation or something else to measure co-localization. But typically it's performed by comparing clusters. So we have localizations that might cluster around each other. And there are also very nice ways published and analysis routines published to perform this analysis. I don't want to go into the details, but basically you perform a nearest neighbor search of one data set and the other, let's say a green and a red. You perform this over different radii, and you get a scoring for the co-clustering with respect to the distance from each localization. So that sort of gives you an estimate for co-localization in SMLM. And it's available in Thunderstorm, actually, and there's also another software platform that was developed that's called Llama that I also used myself. That is quite nice for that. So the other dimensionality that comes usually after considering multicolor is 3D, and 3D is also possible. And the only thing that needs to be done is to sort of make the point spread function sensitive to the three-dimensional position. And the point spread function itself normally is not very sensitive to the 3D position. So you see, it doesn't really change much over an area of over a distance, let's say 600 nanometer. It still does change, and there are some people who can or some routines that were shown to be sensitive to that and extract the axial position by looking at the two-dimensional point spread function. But what is often done is to deform it using, for example, astigmatism, which is where you put a lens in front of your camera that sort of deforms the point spread function depending on the position of the molecule in the sample. And you can see that it changes from a so-called vertical or a somewhat vertical orientation into a horizontal orientation and encodes the Z position. Here's an example of this double helix point spread function that I showed you earlier, where you have these two lobes, and the angle between these two lobes encodes the Z position. But just as a reminder, keep in mind that for 3D, you need even a higher localization or a higher labeling density to get sufficient resolution. Okay, so I want to say a few words before we come to the examples about quality control. I mentioned this sort of problem already when talking about resolution, is that you have your sample labeled and you still you need some sort of a measure of how well your imaging performs, so how well your sample was labeled, how well your sample is preserved also, if you have to fix it or prepare it in other ways. Of course, microscopes are very diverse. There are many different ways of performing this. The conditions are different, so there's a lot of parameters that go into an SMLM data set, and it became clear that it's somewhat important to control the quality of the entire procedure. Not speaking of the software, of course. At the end, the software needs to localize, and you know that you just get a localization, a list of localizations. So I said earlier that many people have used microtubules for that, but for microtubules, the problem is that you don't really know how many, or it's harder to know how many molecules you have per stretch of microtubule. So what is now, or what is a very nice alternative to that, and that was published 2 years ago, is to look at nuclear pores, because nuclear pores have a very defined structure. So they have a very defined organization, and they are made up of a defined number of proteins. And they have also distances, these proteins, that are suitable for SMLM. So it's a very nice quality standard, actually. And what was done in this work is that they generated a genome-edited cell line where all of the NUP96 proteins, so it's one protein within the nuclear pore, were labeled with, let's say, a GFP or a SNAP tag, and that allows you to perform your labeling procedure, for example, using SNAP tag labeling, or by using an anti-GFP nanobody or antibody. And then you have a defined control sample to challenge your favorite microscopy technique. And you can see here they did this with a number of different techniques, also with Storm and Palm and SMLM. And the beauty of this is really that each of these NUP, they're genome edited, so they should all of the copies of this NUP96 within one nuclear pore complex should be labeled and visible. So you can do this in wide field by just looking at the brightness of these things, and you can also do it in SMLM by counting corners or by counting the localizations within one corner to get really a high precision of counting of the nuclear pore, and if you don't, and this number that you get at the end tells you what your localization and what your labeling precision is. So if you don't see all of the copies, that means that you don't label all of the copies, or that you don't localize all of these copies. So that is really a nice quality control in terms of labeling density, and also in terms of resolution. If you can't see the nuclear pore, then you have maybe a problem with your localization or with any form of correction afterwards. Okay, so now in the second or the last third, I want to talk about applications actually. So that was a lot of practical considerations and a lot of technical stuff. So now let's look at a few examples. So one thing where I think SMLM is very strong is structural biology. It's looking at structures, and I've done this myself to some extent. And here are a few examples of structures that were resolved, let's say, using SMLM. The bacteriophage is a very small example. The TORC1 structure and nuclear pores as well as the centrosome. So sort of like multi-protein complex, small multi-protein complexes that are diffraction limited. And so they are too small for normal standard microscopy, and they're too big for electron microscopy. So that's sort of the realm of SMLM, where it can really do a lot. And I just want to mention a technique that I've been involved with myself and developed myself. So we developed a way to look at such large multi-protein complexes where you would normally, like viruses or the centrosome, where you would normally use electron microscopy. But as we mentioned in the very beginning, that in SMLM or that in fluorescence microscopy, you have the advantage of putting labels on your structure, which you don't have in electron microscopy. So that means that you have a very high resolution, so you can see individual proteins, but you don't know what these proteins are. So you have basically no molecular contrast. So the idea of this work that we performed a couple of years ago is to sort of bring together the molecular specificity of fluorescence microscopy with the well-established routines in 3D particle reconstruction. So a prerequisite for 3D particle reconstruction is that you need to look at a large number of complexes, ideally in different orientations. And this was co-developed by the time a high throughput storm modality, where we basically were able then to look at the large field of view with a homogeneous radiance, which was not possible before. And this allowed us really to look at a large number of these labeled centrosome particles. So these are isolated centrosomes. And one of these centriolar proteins was labeled here, SEP152. And all of these centrioles here in this example are super resolved. So with one shot, we were able to look at a few hundred particles at once. So without that, particle reconstruction would not be possible, really. So we created large libraries of super resolved particles, like thousands, tens of thousands. And with that, we were able to perform two different analysis pathways, let's say, that are quite useful. And I want to mention them. So one is 2D alignment. And this sort of comes from the stochasticity of these data sets. So if you think about a 9-fold structure like the centriole, if you look at individual copies of a centriole, because we have errors in all different steps of the imaging procedure, for example, the labeling, you might not label all of these, you might not image all of these, and you might not detect all of these copies, or all of these, let's say, all of these lobes of the centriole. So the information is sort of lost when looking at only one copy. So you need to look at many copies, and then you have to somewhat align them to get an idea about the symmetry organization. So you can also just put them on top to get an idea about the radius of this particular protein complex within the centriole. But only after performing a two-dimensional alignment procedure, you get an idea about the symmetry organization. And we also performed something like that on the TORC1 complex, which is not as symmetric, at least not with our resolution, but we were able to see that it is that it resembles a hollow cylinder in cells. So this software you can also find on GitHub, that is able to perform this type of data preparation to perform eventually a 3-dimensional reconstruction, which is the second sort of output in combination or instead of the 2-dimensional alignment. Okay, and this has been done also with nuclear pores before. it was realized that this is an eightfold structure, that you might not see all of these 8 lobes in each individual structure, but if you put together a large number of them, you get a very high, like sort of an averaged structural information of the nuclear pore by looking at a few hundred individual structures here. Another very nice example where they didn't look at the symmetry, but they looked at the radius, is that study where the nuclear pore was labeled with a number of different antibodies against different proteins. And they all label the nuclear pore. Again, you might not see all of these 8 lobes, but by putting them on top of each other, just by center of mass projection, you get a very high readout for the diameter. And they have done this for these different proteins, and they can reconstruct like the position or the organization of this protein complex in the nuclear pore from light microscopy data, which is quite remarkable. Also viruses, I said this in the beginning or a few slides earlier. So this is one multi-protein complex that I also work with myself. So it is also applicable here. So in this study, what was done is that parts of the tegumen complex in HSV1 and herpes simplex virus were labeled. And again, by combining a number of different individual particles, this allowed this routine that was developed here to get a very high, a very precise readout of the radius of this particle. And again, by looking at multiple structures, they could reconstruct or get a better idea of the organization of an HSV1 particle, which is quite nice, and it's not possible with EM easily. Okay, so just a technical note on that, because I mentioned it earlier. Automation is sort of critical if you want to perform these sort of high throughput, high content analysis pathways. So one way to make it high throughput is to increase the field of view, which we have done earlier. And the other way is just to automate your imaging procedure. And the reason is that SMLM in general is quite slow. You have to acquire a large number of frames at a certain integration time, which just takes time. One way to do this is just to program your microscope and tell it, please image 50,000 frames of that field and then that field and so on. So you just automate it. You can combine this also with structure and pattern identification and all sorts of things. One thing to keep in mind is that, of course, the microscope needs to be stable to a large degree to image for minutes, hours, or even days. The buffer is also something that is important, and the sample might also degrade. So it's not, of course, many microscopes nowadays are automated or can be automated, but that's not the whole story. You still need to take care of your sample that needs to be stable for an extended period of time. Yes, of course, for live cell imaging, UV excitation might be phototoxic, so you also want to maybe automate this and make sure that you don't kill your cells while imaging them. So what comes out of automation is that you can simply look at many or large samples. So many, I've shown you an example of the many part. And in this case, they looked at large samples. I think this is animated, yes. So by just sectioning or looking at different parts of a really big a neuron, in this case, they could reconstruct this entire cell, so which is quite nice. It's a quite nice example, I would say. So it just gives you more another dimension. You can look at larger samples or many more samples. Here's another example where the cell cycle was reconstructed by looking at one divisome protein in Caulobacter, which is a bacterium. And in this case, they did not look at the entire cell cycle in one cell, but took snapshots of individual cells in an automated way. And with outlining or with identifying the cell outline, they could overlay these different structures and reconstruct the organization of this divisome protein over the entire cell cycle. Okay, which is quite nice. Of course, other structures have also resulted in very nice, discoveries using SMLM. The cytoskeleton is studied still using this technique, which is very nice because it's let's say a structure, a cellular structure, as compared to these multi-protein things that I mentioned earlier, that can be very nicely studied with SMLM in combination also now with electron microscopy and correlative studies. So there are very nice examples of that. Another structure that I want to mention is the plasma membrane, which has been studied also using SMLM in T cells, for example, or in other cells, or by using, this is even using what's called light sheet microscopy, so to reconstruct the organization of, I think, a plasma membrane receptor over an entire cell. So that's also something that is very, these are very nice examples of SMLM in structural cellular biology. So I want to mention say a few more works about this membrane organization, as it's also what I'm doing myself. So what has been known for a while now, or shown on many different using many different techniques, is that the plasma membrane is sort of compartmentalized. So it's not a homogeneous, randomly diffusing accumulation of proteins and lipids. But there's a certain organization. The organization comes either from lipid domains. There are these lipid rafts that are debated or that are discussed every now and then. And then there's the cytoskeleton, and there's of course a three-dimensional organization of the cell. All of these components together lead to a compartmentalization of the plasma membrane. And to see that or to analyze that, clustering has been developed, or is sort of the one of the most or typically used techniques to analyze these data sets. So if you look at the plasma membrane, let's say in this case, or in all of these cases here on this side, you get let's say a two-dimensional representation of the plasma membrane, or a three-dimensional, and you get clusters of localizations because the plasma membrane has this sort of compartmentalized organization. So one way to extract this information from your SMLM data set is to perform a clustering operation, which is not, it's also something to think about, because there are many different ways to do this. And there's also been a webinar about this, which is sort of similar to this localization microscopy challenge that I mentioned to you earlier. So this is also something to visit. There's also a number of videos available here, because there are different clustering algorithms, and they have their pros and cons, and that's something to consider when clustering your data. So in this case, they also performed a challenge, I think, using a synthetic data set, a ground truth data set, and just challenging different clustering algorithms and see which one performs good or bad in certain situations. So one that is very often used, because it's quite easy to implement, is density-based clustering, where you just look at their nearest neighbor distances. So each localization gets a radius around it. This is one parameter. And then you count the number of localizations within that radius. That is a second parameter that's required. And then each localization can either then be a part of a cluster or not. So that's quite easy. Tessellation is another one, which is based on Voronoi diagrams. And Voronoi diagrams are sort of polygons that separate localizations always by lines to make equally sized areas, equally sized polygons, and that can be used to identify clusters. And then there are more, let's say, global ways of doing this, by just making radial correlations, as in these two cases, which gives you a global representation of the spatial organization of your entire data set. So some of these really give you sort of an idea of how your molecules are organized, and some others really give you molecule clusters, so they put each localization into a cluster or not. So just one example, one recent example that I found very nice is a paper where they looked at protein complexes in T cells, and this is a DNA paint study. So it's an exchange paint, so it's an extension of normal, or let's say of single color paint. And I mentioned this earlier, so you can just wash out your imager and insert another imager, and then one after the other image a number of proteins of interest. And in this case, they performed a two-step clustering or counting procedure. One is first to cluster your molecules of interest using a density-based, or no, they used Bayesian-based clustering here. So that's a different way of clustering to identify these complexes. And then they use the k-means based. So this goes from here to here. And then they use the k-means based identification of clustering to k-means based identification to identify molecules, to identify individual molecules, which I think worked very nicely here. And I think it's a really nice demonstration of using exchange paint on plasma membrane organization. So you go from a cluster, so first information is that you know how big your cluster is. So for each of these proteins, you can say how they are organized, and then using the k-means based clustering on top of that, you know how many molecules are organized within that cluster. Now for k-means, it's quite important that that works very well with DNA paint, because it doesn't have, do I have it? Yeah, DNA paint doesn't have this sort of stochasticity and the number of blinking events. So it's sort of very defined, the rate of blinking with which you identify your molecule of interest. So each of these molecules here in a DNA origami structure or in your experimental cluster has more or less the same amount of localizations, and that's important. It's important as important prerequisite for this quantification procedure. Okay, so the last part is about live cell SMLM. So what can we, in terms of, first part of, let's say, this nanophysiology case study discussion is structure, which I think SMLM is very good for. So to get nanoscale structure organization of your protein of interest. The other part concerns more live cell. So this is an earlier example where Paxillin was studied, because SMLM is to some degree, of course, live cell compatible. And in this case, they didn't localize each individual, but they put it together into sort of chunks and track a protein organization over discrete time steps. So you can image this. This Paxillin protein was here imaged using Palm. And instead of reconstructing each individual frame, they construct a certain number of frames to get a snapshot of over a few seconds, let's say, of that Paxillin structure, and then go if this same bin further, you get another reconstruction of this Paxillin structure. And it's a very nice way of performing live cell super resolution imaging, I would say. So that's a nice example, in my opinion. The other, I think, good or very nice application for live cell Palm is to do tracking, because if you have a living cell and everything is dynamic and in motion, and you localize a molecule or you activate a molecule, it's of course moving as the cell is moving, and you can follow that movement for a number of frames. And it was this sort of property was exploited in this study. And looking at plasma membrane proteins, for example, you can do this at a very high density. So you have your cell expressing a protein of interest labeled with the photoconvertible protein. And you can not only do this with one as we do in SMLM, but with hundreds of thousands of molecules. And you localize them and you get a trajectory. So you get a very high density representation of molecular dynamics, which is, I think, can be super useful for certain applications. So I think that's a very nice application for live cell SMLM. Just two examples of that. So looking at membrane receptors, again in neurons. And so in this case, this was combined with a wide field label of these, now I forget how they're called, these sort of little buttons here on a neuron, and then performing an SBT-palm analysis on this particular structure. You can really identify different zones, different areas, and compare it to the body of that neuron. So that was a very nice, I would say, example of SBT-palm in a biological system. The other example is more recent, is looking at actin regulation in the lamellopodium. So in this case, different proteins of the actin regulation machinery were labeled with photoconvertible proteins. And by performing an SBT-palm study, also again together with a GFP marker for the lamellopodium, just to see the edge, and also to go away from the edge for a few micrometers to identify these different zones of the lamellopodium. This could be then overlaid with an SBT-palm data set, where you can discriminate, let's say, diffusive molecules from confined molecules and immobile molecules. And it's been seen that depending on the position within this lamellopodium, you see an accumulation of, let's say, immobilized molecules or diffusive molecules and so on. So that's a very nice also combination between localization and dynamics to figure out a dynamic reorganization process within the cell. So it's, I think, a very nice example of the nanophysiology or SBT SMLM for the nanophysiology. So just an extension of that is to combine these to combine a large number of these molecules, and this is something that's been called U-paint. I don't have it here, where a soluble protein was used. So it's a combination of paint and SBT-palm tracking, where the affinity probe, let's say an antibody or something labeled, is added to the surrounding medium, and you can label your membrane receptor with that probe for a certain number for a certain time and track it. And this led to very spectacular representations of the diffusivity of these proteins, and it can be translated from, let's say, this tracking organization into a pixelated visualization of diffusion coefficients. So you really can finely map the diffusion properties of your protein and relate it to the position of this on the cell. Okay, so that's something that I could also demonstrate, or that is available in this tutorial GitHub wiki that is analyzing SBT-palm trajectories or even in general particle tracking trajectories. One general analysis that's often done is to perform a mean square displacement analysis. So where you have you localize your particle, or you localize your single molecule, and it goes from here to here, and each of these individual steps is associated with an XY pair of coordinates. And the basis of MSD, of the mean squared displacement, is to take the lag time, and for each lag time, so for each time step, let's say 1 to 2, 2 to 3, so for one time step to 2 time step, and so on. So to compare this displacement with respect to the time step, and this allows you already to identify different modes of movement. So, for example, if it's a normal diffusion, it would be a somewhat linear relation because of the way it sort of the particles move. If the particle is confined, of course, it will hit an invisible boundary at some point. So this is very common when looking at live cell trajectories, that the particle will not move away even further, depending on the time lag, but it will sort of hit a boundary and then don't move any further. So that's also something that's very useful when looking at live cell experiments. And then, of course, there's all sorts of things in between. We have anomalous diffusion or active transport. So one thing that I want to mention that is also available in SPT Palm is to look at the lifetime of clusters. So we have discussed clusters before. So if you do that if you perform this clustering on a live cell Palm data set, you can extract the number of clusters, or you can extract a cluster using one of these routines that I mentioned before. And then by looking at the frame number or the time at which each localization appeared within that cluster, you get an idea of the lifetime of this cluster. And if you put this in a cumulative plot, as in this case, you really get the minimum lifetime of a cluster, let's say. So let's say this blue line here, really this a single cluster that was identified using, let's say, density-based clustering. And then you can count the number of localizations. And this cluster accumulated localizations over a time of 400 seconds. So you get the lifetime, and you can discriminate the organization of the cluster, the dynamics of the cluster. So you get a very rich it's a very rich type of data set, I would say. Okay. Okay. So this is the part that is described here in the wiki, and I could also click through that if you are interested. Just one note that which is a type of software that I find is really useful for representing this type of SPT Palm datasets, called Inference Map. I think it's also mentioned in the software category. Okay, so just one last point. I will I will present some of my own data later this week on Wednesday, where we'll give a few examples of SMLM in understanding virus biology. For example, using virus tracking, using SPT Palm, and also clustering, even combined with some simulations. So feel free to pass by there. So just a few take-home notes. So one thing I think that is important to consider with any super resolution technique is really to first sit and think if it's really necessary for your application. SMLM then again might not be the best technique for your specific question. So there are other super resolution techniques, of course. Then if you decide to do that, what you would need is, of course, an instrument to perform these measurements. You need a sample that is labeled in a certain way, and you need to familiarize yourself with data analysis. Samples still need to be, because you look at individual molecules. So if the sample is not good, it's going to create a problem at some point. Labeling should be optimized. Labeling size should be taken into account. That's something that I mentioned earlier. And the imaging conditions, of course, are also important when performing these nanoscale measurements. So for some, you need total internal reflection. You need to, or maybe it's not the first thing to do, but optimize the switching kinetics, which is something that I mentioned earlier when taking care of the blinking. Stability and drift correction is usually important to consider when designing a microscope or performing corrections on single molecule localization microscopy data. But of course, what you then get is a very high precision data. So you can localize or pinpoint your molecules at very high precision, on the order of tens of nanometers or even below a few nanometer. So that's quite spectacular. If you have good labeling, you can get very high spatial resolution as well. It works on live and fixed cells in general. And applications are more suitable for one or the other, as I try to point out a few times during this lecture. And then, a lot of time should be investigated into extracting quantitative information, and that's really something to spend an entire lecture on. And I try to make a few points in this nanophysiology part, but there's many different ways of quantifying SMLM data sets, which is and that makes the technique extremely useful in quantitative biology, I would say. Okay, so if you want to know more, feel free to browse these sites. We can also have a chat now, so I'm going to stop my screen sharing so that we can see or go through a couple of comments. Or as I said in the beginning, if you want, we can go through 2 points, let's say the photophysics point and the tracking point in a demonstration.
Please don't stop sharing your screen.
Okay. All right, so I leave it like this.
Okay. So, thank you for your very impressive presentation tutorial. I haven't seen questions in the chat yet, but please ask if you feel. But I can start with a question. So you said that you can do live cell imaging, but I still have some problems coping with that. You say that to have a nice structural image, you need something like 25 minutes, correct?
Yes.
So in that time, the cell has been moving.
Yes.
So how can you then, with an image of 25 seconds, let's say, have a good view of the structure?
So there are different ways of viewing this. So the Paxillin example, for example, the Paxillin example was something where depends on at which time resolution we want to look at the structure. So in this case, they looked at the large structure, which is the focal adhesion. So they took images. So you can take images at very high, very low integration time. I don't know, 30 milliseconds. So and you reconstruct one time point by using a number of frames. For example, a few seconds of frames, let's say 5 seconds. So then you have the structural organization snapshot of these 5 seconds. And you can do the first 5 seconds, next 5 seconds, and so on. And of course, the sample moves. But if it's a larger structure like the focal adhesion, it might be very well suited for that. The other time regime, I think, that is suitable for a live cell SPT palm is the very low or the very fast dynamics, the very short time scale, which is the regime of SPT palm, where you look at the dynamics of individual proteins.
Mm-hmm.
They move on a matter of milliseconds to seconds, and since these trajectories are very short, we can just accumulate trajectories over a longer period of time. Of course, the entire cell might move, which is important to take into account. But to really directly answer a question, so of course you cannot reconstruct a complex. Let's say if the complex is in a moving cell, but if it's a dynamic complex, you have at least some time resolution by doing these time bins, let's say of 5 to 10 seconds, as in the focal adhesion case.
So if you, for example, would like to follow actin, moving all the time, that will be more difficult because it's smaller and you need longer times.
Yes, exactly. I think actin could be difficult to follow, and so you would just not get a complete data set. I mean, you can get a snapshot of. I try to remember if there's been an example of this. But actin has been combined, for example, with SPT palm in wide field by just using something like live act. So you get a snapshot of actin and you overlay it with your SPT palm trajectories. And you can get an idea if your movement is correlated to actin. But yes, to reconstruct a small structure is going to be difficult because it's going to move at the same scale as it is organized, right? Yeah.
And then I have thank you, and I have a question here in the chat. Thank you for the nice presentation. The first sentence is, in ratiometric imaging, how are you taking care of crosstalk, as they are very close spectrally?
Yes. So actually, you want crosstalk. So you use the same. You should use the same laser irradiation, and these molecules appear in either channel. And what you do is simply to take the ratio of these two. So you want this type of crosstalk, so to have them so close by and to appear in each other's channels, and you simply take the intensity ratio of the two. Yeah.
Okay, so I hope that answers the question. If not, let us know. There's another question. Wondering if you know of a good algorithm to measure length of tubular structures from a D-storm data set that are flexible, it is not rigid.
Yes, so there's two examples that I know, but now of course I can't come up with the name. So I think yeah, so there's one paper that came out a few years ago, I think in molecular cell or something like that was used to identify tubular structures, so just to extract them. I don't know if it gives you the length. It's sort of, I think it was sort of a network, but you can just maybe send me a message and I can try to find out, or give me your contact. So this was sort of a network analysis paper where they looked at overlapping. Of course, they overlap at some point, and they looked at this networking organization and had some readout there. The other paper that I remember now is something that came out of Dylan Owen's group. Who is, they're really specialized on clustering algorithms and clustering research, and I think they published or they were working at least on something to extract tubular clusters, so from microtubule data sets at least. So that would allow you to just extract these features. Then to measure the feature is a different question. So you would have some way to I guess to find the outline, fit either some sort of angle or shape to it and measure it. I think there was another paper from Markus Sauer. They looked at this synaptonemal complexes. That might also be worth looking at. And I think they had some way to quantify these synaptonemal structures by doing, I think, just that. So extract it first somehow, then segment. So you need to segment the tubule, let's say a tubule between like crossings or so, and then perform some sort of analysis. So these are the things that I can come up with now.
Okay, thank you. I have another question myself. You say that you can do 3D imaging with SMLM. How deep can you get into a cell, for example, from the cover slip?
So that's very specific. I think there's normally not so deep. So with these sort of principal ways that I mentioned, because the point spread function is going to distort at some point, but there's ways to overcome this. But now, I don't have the specifics now, but I think there's a couple of implementations to do this deeper inside the sample, but I can't give you any specifics. I think it might be worth looking at the papers from Sandrine Levesque-Fort from France. I think they have developed some ways to look deep into the sample. Of course, you can correct it. That's another way. You have to get some way of correction. So I don't know, do this astigmatic type of calibration of beads that are deep inside the sample, like they are embedded into some sort of matrix. But of course, it creates problems as soon as you go, I don't know, a few micrometer deep into the sample.
Yeah. So you're limited to a few micrometers, basically. So basically, it should be very close to the cover slip.
That would be the best performance, I would say. Yeah.
Okay, thank you. I don't see any further questions. So, Christian, it's up to you what you want to do now. You want to continue?
So I'm just going to stop this. Maybe I just, so if we still have some participants in the...
Yeah, we have all the participants still.
I can just show you one thing from the tutorial wiki, which I think is maybe useful for some people. So let me stop my screen share, or at least start a new screen share. So now I'm gonna share my entire screen. I think that's easier. Okay.
Yeah.
So if you go to the wiki, that's just, this is something that you will find. And I just want to maybe click through these because that's something that is quite useful. I had prepared these two, the tracking and the photophysics, but since I was asked for this a few times, maybe it's worth going through. So here,
Christian, can I shortly interrupt you? There's a new question. Yeah, this is really live. To answer the question from Mark, is it possible using coating like gold coating or silver coating cover slips to get deep into cells?
Well, are they then still transparent? So I don't know if that would not interfere with like the normal optical identification of your sample. So I'm not really sure if that would solve the problem. Yeah.
Maybe
if I understand.
I don't know either. The question is not for me, but maybe the person that asked the question. I could give broadcast permission if you want that. So then you can take the floor yourself as a question and explain. Do you want that? Just type yes if you want. There's no yes. So
it might well be. I'm
not familiar with idea. He doesn't have a
I'm just not
suggestion. Yeah. Okay, thank you.
Okay, so let's just go back to the wiki and so where was it here? Okay, so then the way to perform this type of so if you want to perform a grouping or a counting operation using SMLM, so I'm just going to click you through what you will find here on the wiki if you want to use it. I have to say that most of the scripts that I provide here are written in MATLAB, and that might just excite you or not right now, but this is something that I have used a lot. I tried to get into a few others, but it's still MATLAB for most of what I'm doing myself. So in the photophysics, so if you click here on the photophysics part, you will find 3 scripts, and now I go to MATLAB. No, I don't. Where is it? It's crashed. Okay. MATLAB crashes when I disconnect my second. No, I have to start it again. It just has problems with secondary displays. Okay, so I also provide a data set here. So let's click. So if you go to the photophysics, you find this photophys hat, grouping and analyze, which are the ones that are important right now. And in the tutorial, on the tutorial sites, there are also some example data here to perform these steps that I just want to describe. So if you go to the photophys hat, the first thing that's going to do is to load one of these data sets. And this is the one that I just described. So it's really just a fluorescent antibody spotted on a glass slide and imaged at conditions that are very close to your experimental conditions. So if you load that, it's not going to work. Oh, yes. I forgot one. Okay, so here's a representation of that. So it's really not very spectacular. There's nothing in it. It's just fluorescent molecules on a glass slide, and I can try to zoom in, let's say, on an area here. So you see that each of these individual molecules will create a cluster of localizations, as in this case. And what we want to do with these scripts is to analyze these clusters, like to extract the scattering and the blinking properties. That's what this part photophysics does. So the first thing that we want to do is to, and I'm using tracking here. So a simple tracking operation really looks, it starts with each localization, and it goes in step and steps towards the number of frames. So it goes down the number of frames and looks for other localizations that are within a certain distance radius. So that's a very basic tracking operation. And in this case, I perform this tracking operation with a gap time of 0. So this is supposed to combine localizations that are in one blinking event. So if you remember this chymograph that I showed, so if these blinking events can come in subsequent frames, so frame 1 to 5, let's say, is one blinking event, and then the molecule goes off and comes back in frame 500. So what we first want to do is to combine these blinking events. No. Oh, yes. I have to, so some of these functions are here in the common function part because I had to restart. Okay, so first we track with gap 0 to combine blinking events. Then we merge them into grouped molecular positions in the second part, and then we have to track again with the gap time that is set to the maximum number of frames, because so now we have blinking events for each molecule, and we want to now identify each molecule. So we want to know how many blinking events we have per molecule. So we track this data set again using the gap time set to the maximum imaging time. So what is also important then is during these type of calibration experiments is to set the time also to something that is realistic. So don't image too short to just get a few blinking events. So typically what I've done is to image as long as you can see anything, basically. So because these dark times, as I mentioned in the presentation, they can be very long. So they can be tens of seconds. And to consider that, really, take a very long data set, even if it's going to be largely dark, just to make sure that you get all of these, you get the very long off times. Yeah, so of course this part takes a bit longer because you have to follow each molecule until the very end of the acquisition. But you can already, I can already go to the analysis part. So the analysis sort of gives you, tries to quantify these different aspects. And the grouping part is really then just to perform a grouping operation on your data set. So I'm not going to click through that. It's quite short. It really only loads the data, performs the grouping, and merges the localizations. But to perform this type of analysis, you need to go through these analysis steps first to get the required quantities. Still busy.
Okay.
Yeah, just a second. Actually, no, let's just wait. And I think it's also going to, so if we go back here, these steps are also discussed here, these different outputs, but I still would like to show you if it's going to give us the result at some point. Okay, well, let me see if I can keep an eye on the busy sign. So, okay, so one of the first things that comes out is the on-time distribution. So if you track with gap 0, you combine blinking events, and the length of each blink event is sort of the on-time, which is the first sort of important parameter, because you want the on-time to be around the integration time, right? So that's something to keep in mind when optimizing an SMLM experiment. So often, so to get the maximum number of photons, you don't want to distribute a blinking event across too many frames. So one way to optimize this is by performing this operation and reducing the on-time all the way down to maybe 1 or 2 frames. So that's the first output. The second output is the number of blinking events. That is just a property, sort of an intrinsic property of your an intrinsic property of the dye. Yeah, in this case, the dye, and of course the imaging conditions. And in this case, I used Alexa 647, which blinks like 7 or 8 times on average during a typical acquisition. The next part that comes out is the dark time. This is what took so long now. Now it's actually done, but never mind. So the next time is the dark time distribution. And I plotted this in this way. As you can see, that the dark time can be actually really long. So maybe its mean is somewhere around 80 frames. This is frames now, but the molecules can be off for a very long time. And then again, to have a high confidence on merging all localizations of an individual molecule, as in this case, I set it on this 99% quantile, which is around 1300 frames in this case. So this is one of the critical parameters that you would put into the grouping procedure. The next part is the localization precision, which is also very important to know. How precisely can I localize molecules in my data set? And in this case, there are 2 ways of doing this. One is shown here, to put all of these individual molecules on top and performing an average fitting using just a normal distribution. And it gives you, or in this case, resulted in a lateral localization precision. This is a 2D data set of like 15 to 20 nanometers. And that's something that we also put into publications, for example, because it's an important parameter to know if you, for any subsequent analysis, if you have a too high localization precision, it might just compromise your quantitative information. And that's like, this is an output that also comes out from these scripts. So the lateral localization precision, then let's say 15, 20 nanometer. It's going to be larger in 3D. I don't have it here, but in 3D, it's typically around, let's say, 30 to even 50 nanometer. And so these 2 numbers you can then put into this grouping script, and even like as a test, reanalyze this test data set, and instead of localization clusters, you should then see only individual molecule positions. Okay, and that's basically one way to. Okay, now I don't have to click through this anymore, but it's actually one way to perform counting on STORM data sets. Okay. Yeah, that's it, I would say, from my side. Let me stop the screen sharing.
In the meantime, there was one question: what is the time frame from excitation through dark state and back? But I think that you answered that question in this part.
Yes. Yeah,
so it can be very long.
Yeah, in this case, it was like on average around 80 seconds. But for STORM, it can be very long, which is also a problem for STORM, counting in STORM. And I didn't mention this, but there's other ways to perform quantitative SMLM using, for example, paint, which is better to control, as you have these very defined blinking kinetics that can be calibrated using, for example, DNA origami. Also, PALM fluorophores typically don't have these long dark states. So PALM has also been used very successfully for counting. But basically, you can get it from all of these techniques as long as you calibrate your blinking kinetics one way or the other. Yeah.
Okay, thank you, Christian. If there are no further questions, and I haven't seen in the chat or in the question mark, then I would like to thank you for your very clear tutorial. And it's time for a little break until 11:30, I think, when everything is opened officially. So we all hope to see you back then.
Yes. Yeah, thanks, Mark, for the support and for keeping an eye on the chat. Thanks, everyone, for attending. And if you have any questions, just feel free to contact me. And enjoy the rest. Let us enjoy the rest of the conference.
Thank you.